Good morning, everyone. Thank you, Lee, and thank you to Aspetar for providing me the opportunity to speak to you this morning. I'm going to talk to you a little bit about the work we've been doing recently on metabolic alkalosis, specifically, and neuromuscular function in the context of fatigue and fatiguing exercise. Now, when we look at the association between skeletal muscle fatigue and exercise performance, if we relate that to a decline in pH, or oftentimes referred to as metabolic acidosis, the two major players here really are metabolic in nature and contractile function. In terms of metabolism, really we're talking about the anaerobic pathways here. Specifically, acidosis is affecting glycolysis, glycogenolysis, some other factors associated with anaerobic energy provision like phosphocreatine and inorganic phosphate, and also something called strong ion distribution, which is simply referring to some of the stronger ions, such as sodium and potassium, and how they're distributed across the cell membrane. In terms of contractile function, oftentimes metabolic acidosis is not linked to maximal force, but rather the impacting negatively upon velocity of contractile shortening. Specifically, we're talking about inhibition of some of the weak to strong binding states, and also been linked to a fiber type dependence, whereas acidosis seems to be affecting the fast twitch fibers more than slow twitch fibers. So one of the challenges that we've tried to overcome anyways is a lot of the research in this area, specifically on acidosis and the specific effects on contractile function, has really been on isolated muscle preps, either single cell, single fiber, or again isolated muscle after dissection. And so what we've tried to do then is translate some of these findings and see whether or not we're able to measure in an exercising human accurately whether or not the same findings and results that we're seeing in isolated preps are the same and can be translated ultimately into practice during exercise. So what we've had to do is take much more of an integrated systems approach, and especially important when you're looking at fatigue in the context of this type of exercise. So if we look at a decline in pH or metabolic acidosis a bit more thoroughly, like I mentioned before, we've got our metabolic effects of a decline in pH, where we have an effect on glycolysis, glycogenolysis, as well as some of the other factors associated with anaerobic glycolysis. We also have our velocity of contractile shortening. But again, like I mentioned about the strong ion difference, some of these changes that we're seeing across the membrane when an individual becomes acidotic can affect the rate with which the action potentials are propagating down the membrane, ultimately affecting probably the rate of velocity here. One of the measures that we've started with, and we've had some success with recently in an applied research sense, is the rate of force development. Rate of force development gives us a picture here of what's going on at the level of the actin and myosin interaction, but also is influenced by some factors up above and beyond that neuromuscular junction. So how do we go about measuring rate of force development? Well, remember, it's an explosive contractile estimation of power, really. And probably the most reliable way we can do this in the lab setting anyways is through producing a maximal voluntary contraction. Now, how do we go about doing that? Well, in this example, we're talking about an isometric knee extension model where the subject is asked to push against an immobile lever arm here, where we have a force transducer here. And if we look at what sort of force traces we get, obviously at rest down here, when we ask the subject or individual to push as hard as they can, we see an increase in force, or the force trace here, up until they've reached their maximal voluntary capacity. We ask them to hold for 3 to 5 seconds, and then obviously relaxation here. What we're specifically focused on, though, with this rate of force development measurement is this time period right here at the initiation or the onset of contraction. Oftentimes we double check with an EMG trace, but simply it's just a measure of power and the onset of contraction within the first 200 milliseconds or so of the contraction. So it's a real immediate and a real finite measure, oftentimes very difficult to measure accurately. It requires the participants to be well trained and well familiarized with the activity that you're asking them to do. So our first study that we put together, really going back to that issue of difficulty in measuring rate of force development, we wanted to get an applied model, so we wanted to have something with a bit of ecological validity, such as riding a bike. But the problem with measuring rate of force development is that it's really impacted upon by, or I should say, fatigue is very transient, and it goes away quickly. Okay, so the measurement is very difficult. It has to be done almost instantaneously. So one of the problems that we had originally was using that dynamometer example that I showed you a minute ago, is that by the time we asked the cyclist to ride at an intense rate, get them off the bike, strap them into the dynamometer, we weren't seeing any changes. So we had to adjust our model here, and this is just simply a picture of what that looked like. We just cut an old sit and reach box in half. We attached 2 force transducers to either side of the pedal, okay, and we were able to slide the box backwards and forwards. And so what we had them do was exercise. When we asked them to do a maximal voluntary contraction, then slid the box underneath the pedal. What you can't see here is that the cyclists are also strapped in and harnessed to a squat cage to eliminate any counter movement. So again, looking at the effective pH on rate of force development, we initially altered pH just through a standard technique of ingesting sodium bicarbonate. Gives us a significant elevation in pH. We then had a standardized warm-up, but our exercise trial here was intermittent in nature, very highly intense, 120% even of their max, and they rode for 30 seconds here at that pace. Immediately upon stopping, we shoved this box back into the underneath the pedal and measured a maximal voluntary contraction. They had 20% of an active recovery for 30 seconds and then repeated the activity. So 30 seconds on, 30 seconds off, 30 seconds on, 30 seconds off. And each time we measured MVCs until they could no longer go or maintain the 120% output. So a time to failure task. So looking at some of the individual data here, again, going back to our force trace here, remember that if we ask somebody to do an MVC, we're going to see a trace that goes up, and then it's going to plateau. And this black line here on the left and the gray line here is just simply transposed down into this small 100 millisecond time frame down here. So what we can see and what we started to see over the course of the fatiguing exercise is that the alkalosis condition, or inducing a pre-exercise alkalosis, really attenuated the fatigue process. So what we're seeing is less of a decline in the slope, so less of an impact on the rate of force development as they got more and more fatigued. And if we look at that over the course of the exercise session, so this is everybody included, all 10 subjects, and all I've done down here on the x-axis is normalize the time to failure. So everybody obviously will fatigue at different rates. So I've just normalized it to 100%, and then broken down the nearest MVC to 20% effort, 40%, 60%, 80%, and so on. Over here on the left, we've got normalized RFD, again to max, so obviously starting off at 100%. But what you can see all the way through this fatiguing cycling exercise, upwards until we get to 100%, is an attenuation or a reduction in that loss of explosive force capability when we make an individual alkalotic or when we raise pH. So this really made us look at this in a bit more detail and really try to explore what the mechanisms are that are responsible for this relationship between a pH and rapid power development. Okay, remembering here that our model is integrative, and knowing that rate of force development is impacted on by the neural aspect as well. So we began to develop a model here. So we have rate of force development, where we've seen an increase in the rate of force development, okay, with this particular study here. But knowing that there's an aspect of motor involvement, we wanted to look at a couple of other studies, specifically this one here, looking at voluntary activation, afferent feedback indirectly, which I'll talk about in a minute. But again, also whether or not we might be able to measure a fiber type dependence in these individuals. So just briefly, for those of you who aren't aware, there are a lot of different ways you can measure central contribution. One of the methods that we've adapted in the last couple of years is the twitch interpolation technique, which just involves a stimulation, a supramaximal stimulation, either at the brain level of the spinal cord, or what we've been doing recently, in more at the level of the motor unit. And all you're trying to do is maximally stimulate the available motor unit pool, and you're able to maximally recruit. And so what we have here on our force trace, this is our typical force trace. Remember, you've got an explosive movement here, you've got a plateau in maximal voluntary force. If you superimpose a stimulation here, like we do, in this example it's on the femoral nerve, if you superimpose a stimulation, you're able to look at whether or not there's any involuntary drive left to the muscle, okay, that you're unable to recruit. So we look at that, and then obviously we normalize that to what we call the peripheral twitch, which I won't get into any more specifics on that. But that's the method that we've used. In terms of the first study, remembering that central drive is a key component to muscle contraction in an exercising human model. We looked at trying to isolate it a bit more, and so we used a supine calf plantar flexion model where our subjects exercise for 3 seconds at 55% MVC, and then they relax for another 3 seconds. And they did that intermittently, again, till task failure. Okay, so very isolated, not whole body, but isolated in this instance. And we tried to see what the range of pH would have, whether or not the range of pH would have an effect on this. So we looked at an acid condition, ammonium chloride, and then we implemented a basic condition, again, sodium bicarbonate, and then they had a neutral condition as well. I'm only going to show you the alkalosis findings real briefly. So remember our superimposed switch here, and this might be slightly difficult to see, but it is raw data, so it's a single subject data. What we see at the beginning in a non-fatigue state here, on this superimposed switch, it may look a little bit different, but it's not different, I guarantee it. Our superimposed normal pH and our superimposed alkalotic pH to start with. What we've seen over the course of the fatiguing exercise is a dramatic increase in that superimposed twitch in the placebo condition, where we haven't altered pH. Whereas in the alkalotic condition here, we see a very small increase in the superimposed twitch. So we're able to use more voluntarily in this scenario than we are here. There's a lot more left over in the muscle that we're unable to drive for some reason or other. Okay? So that's led us to look at a few other things, and this particular model was looking at attempting to indirectly assess whether or not we're impacting upon the afferent firing rates, which is a contributing factor to central drive. So we developed a very, I would say, very difficult exercise trial. This indirect method of assessing afferent firing requires your subjects to be ischemic. And so what we tried to do was, by changing pH initially, we know that when you exercise intensely, you increase the metabolite concentration in any given solution. So we had them exercise intensely for 2 minutes. We had a 2-minute sustained maximal voluntary isometric contraction, a knee extension model again here. And then after 2 minutes, we didn't let them off the chair. We inflated this cuff here for total occlusion and had them sit for 2 minutes while we stimmed them intermittently, which is painful. If any of you ever had total occlusion, especially after you've exercised intensely for 2 minutes, is pretty painful. Alexa's going to talk about pain here in a minute, more specifically. Then at the end of the trial, we went for a further minute. We left them cuffed. We wanted to make sure that they were actually extremely fatigued, and we asked them to push again for another minute. We were able to successfully do that, fatigue them. So again, looking at voluntary activation, when we start off here, we're close to 100%. We never actually get values that are 100%, but we're close to 100% here. We go through our 2-minute periods, or 120 minutes, where we have a decline in both conditions. So both the alkalosis and the placebo condition decline in terms of voluntary activation, but not different between conditions. Once we cuffed, however, we started to see a dramatic difference, and probably more exacerbated than what we saw in the previous calf study. So here in the gray, we see voluntary activation is better maintained, similar to what I just showed you in the calf. whereas in the placebo condition, we have a steady decline in voluntary activation. And this is what we believe is directly linked to an enhanced afferent firing rates coming from the muscle, or an improved rate of afferent firing. Interestingly, though, we see this change in voluntary activation. When we go back to some of our force measures, we didn't see any difference in force or maximal force over the course of this period, probably due to the fact that they were so fatigued after the first 2 minutes. We desensitized the muscle enough to where that we weren't able to tease out any effective pH. But going back to the rate of torque development, or the rate of force development here, which is our central aim, really, to see whether or not we're affecting that. Initially here, we've got very high rates of rate of torque development, or rate of force development at baseline. This is at the end of the 120 minutes, just after 20 seconds of the cuff, so not very long into the ischemic round. We see a similar response, an elevated, although very, very dampened effect due to the fatigue, but we see still an attenuation of the rate of torque development here. But then once we have the cuff on for a prolonged period of time, we don't see any difference between the two conditions. So initially, we see a similar sort of response to what we've seen in the past, but again, the ischemia probably caused too much, and they were too fatigued, really, to see any difference between the other time points. And this would led us to the final study, actually. We started to look, this is a relatively new paper that we've just published. Again, going back to that idea of trying to translate some of the basic science into practice and see whether or not some of that basic science actually can be measured in a human. What we've tried to do in this particular study, because some of the research would suggest that acidosis is affecting fast twitch fibers more prominently than slow twitch fibers, and those studies that do suggest that have all been done on isolated muscle preps. So we tried to replicate that using human subjects. And so we chose what we felt were the best representative muscle groups, a tricep brachii and a tricep surae in the calf, tectonic stimulation protocol, where I'm feeling a bit sadistic, actually, all these difficult and stressful studies, exercise studies. What we did really was we just simply maximally stimulated an individual sitting, say in the triceps model. We had the belly of the triceps stimulated maximally, where we stimulate it, recover, stimulate, recover, stimulate, recover, to try to mimic a contraction. But also what we're doing in that sense is knocking out any central contribution. So we're looking strictly at peripheral fatigue in that sense. And if you look at the calf model here, obviously the axes are different, 160 in terms of Newton meters, twitch amplitudes, and 30 in terms of the twitch amplitudes of the triceps brachii. But what you see is a general pattern over the course of 2 minutes. And we used 2 minutes initially because this would replicate some of the animal models that have been used, but also 2 minutes with your calf being stimulated supermaximally caused a couple subjects to drop out. It was extremely painful and a lot of cramping, induced a lot of cramping. But nonetheless, we continued on. This is the twitch response. It's a generally plateaued response, a little bit declining, but whereas as you would expect, that force production in the fast twitch fibers drops dramatically at the beginning and then just sort of plateaus. So this one wasn't that painful because you were tired after 20 seconds anyways. Intermittently, at the beginning of this, before we started this stimulation, we did a voluntary component, so we looked at the voluntary side of things as well. We stopped at 60 seconds so that the subjects could do another voluntary contraction, and then we finished off at 120 seconds at the end of the exercise doing another voluntary contraction. So we're trying to look at the peripheral contribution outside of the central contribution, but then again, adding both components in there as well. And were we successful? No. pH and fiber type, in terms of our response and our study, the results that we've seen, this is a maximal force over the triceps brachii and triceps surae. So this is baseline. This is just the MVC, the voluntary contraction, again at the beginning, that 60-second point, and then 120 seconds in. Okay, so you're seeing a fatigue pattern. Okay, their inability to produce force maximally is certainly dampened by and so they are experiencing fatigue. But we didn't see any difference in any of the markers between conditions, whether or not there was no effect of pH or altering pH on the fast twitch fibers as opposed to the slow twitch fibers for any of our peripheral or central markers in this instance. So where does that leave us? Back at our model here, rate of force development. Again, we're pretty confident now in three separate instances we've seen pH have a positive or altering pH or in terms of inducing a metabolic alkalosis has improved the explosive force capacity, especially during a fatiguing event. Whether or not that's voluntary activation and more specifically an enhanced efferent firing rates, we don't know yet. The fiber type issue, in terms of measuring it in humans, at this point I'd have to say it's relatively difficult. You could certainly argue that we could use a different model. And we probably will look at a little bit more of an active whole body model next time. But at this point, I'd have to say that we've been unsuccessful in discerning any differences between fiber type as far as pH is concerned. So just to summarize, so rate of force development, or that explosive power, appears to be improved when you manipulate pH, and specifically when you make an individual alkalotic. Voluntary activation, or that drive to the muscle, appears to be influenced positively in those changes in pH. And whether or not those changes in pH are due to enhanced efferent firing rates is left to be determined, okay, and up for future studies. Thank you for your attention.